Next is Adrian Velazquez. Adrian Velazquez Campoy holds a degree in theoretical physics and PhD in applied physics by the University of Granada, postdocs at the Department of Biology in the Johns Hopkins University, Baltimore, USA, and later back to Spain as a Ramon y Cajal researcher. Adrian is an allied researcher at the Institute of Biocomputation and Physics of Complex Systems, BIFI, of the University of Zaragoza here in Spain, and has recently entered the Spanish Royal National Academy of Medicine as corresponding academic member in the biotechnology section. So congratulations on this, Adrian, when you're ready.
Thank you very much. I would like to thank the organizers for inviting me to be here today to speak about some particularities in our day-to-day work. I'm going to talk about biophysical tools for drug discovery, in particular blocking protein-protein interactions in IDPs related to cancer. As it had been mentioned before, drug discovery is one of the main activities at the Institute BIFI. So new medical needs arise because of new diseases, either infectious or genetic, neglected and rare diseases, or aggravated known diseases due to resistance, low efficacy of current drugs, and so on. So through drug discovery, identification of the new drugs, preclinical tests and clinical tests, followed by drug development, new drugs need to appear in the market. We are convinced at BIFI that academic labs are able to go from validation of target molecules, in particular protein targets, to in vitro assays, experimental and computational screening, then the optimization iteration process, and then preclinical assays just before going to clinical trials where the participation of big pharmas are currently required. We are able to use specifically tailored screening processes where we can use either specific activities of the protein target or general properties of proteins in general to implement screening procedures, efficient screening procedures to find new promising drugs or potential drugs. We have been studying different protein targets. Our main strategy is to study, using biophysical protocols and methodologies, the protein targets, so obtaining useful information about the structure and the function of the protein. We are able to identify compounds, small molecules, able to bind, to interact with the protein target. We have been working, for example, in NH3 protease from the hepatitis C virus, flavodoxin from Helicobacter pylori, human phenylalanine hydroxylase associated with phenylketonuria, and human NUPR1 associated with pancreatic cancer. The main basic idea is shared by infectious disease or genetic disease tackling. For an infectious disease, we just need a small compound binding to a key protein in the infectious microorganism and blocking a key process, key metabolic process in the microorganism. For a genetic disease, either for a protein that is showing defective activity or is showing too much activity in our body, we just need also a small compound interacting with the target protein and modulating the activity of this defective protein. So we just need to identify, we need a way to identify compounds binding to a given protein. Later on, we can study if these compounds have toxicity, side effects, if they show activity in cells, in tissues, and finally in organisms. Pancreatic cancer is one of the most aggressive types of cancer with only 6% 5-year survival rate, 6 months median survival, and is the 4th leading cause of cancer-related death in the US. And this is also shared by all countries worldwide. We focus on NUPR1 because it's a protein involved in pancreatic ductal adenocarcinoma development. It's overexpressed in acute pancreatitis and in precancerous lesions. It controls or is associated with cell migration, invasion, and adhesion. And it also interacts with chromatin, interacts with many protein partners, and is associated also with chemoresistance. So NUPR1 is a very good, in principle, very good protein target for PDAC, pancreatic ductal adenocarcinoma. This protein target has is associated with with different challenges. So NUPR1 is a multifunctional protein. So if we block NUPR1 interaction, we may block beneficial and not beneficial function, deleterious functions. NUPR1, as I said, is interacting with many other proteins and with DNA. We had to block protein-protein interactions. Protein-protein interactions are very hard to inhibit with small compounds because protein-protein interactions are characterized by large interaction interfaces, and with small compounds, it's difficult to block the many intermolecular interatomic interaction established between one protein and the other protein partner. And NUPR1 is a disordered protein. It has no secondary structure. So we cannot use tools like X-ray diffraction to obtain information about key structural elements in NUPR1. So we had to rely on computation, molecular simulations, and other biophysical tools like NMR to obtain information about how NUPR1 interacts with other binding partners. So we use a molecular screening procedure to identify compounds binding to the protein, and then we had to use secondary screening procedures to select those compounds specifically binding to that protein, and then not being toxic and not showing side effects, finally in cells and finally also in mice. So this is a multidisciplinary project where we had to use biophysical tools, cell biology, animal biology, and computational tools to tackle the problem. We selected finally 15 compounds that were identified through the thermal screening assay. We used many tests to show that these compounds, or some of these compounds, were specifically binding to the NUPR1 protein. For example, we did PLA assays to show that the presence of the compounds inhibited the interaction between NUPR1 and MSL1 protein, that is a natural partner, binding partner of this protein. And at the end, we were able to show that in mice, in xenografted mice with human tumors, pancreatic tumors, one of the compounds, compound 15, was able to completely arrest tumor growth in mice, compared to the control group with no treatment, where the tumor was increasing continuously, even exponentially. And I wanted to make it very brief, because I know you are quite tired. I want to thank main collaborators in this NUPR1 project and other associated projects. Olga Avian, that is in the audience, from the IACS in Zaragoza, Sonia Vega from BIFI, Javier Sancho, who talked before about Lacrima facility, Angel Lanas, who is also in the audience, and collaborators from other places, José Luis Neira, who is an expert in NMR, biological NMR, Juan Yubana, who is the father of NUPR1 protein. He discovered the protein, and he led this project from Marseille. And Bruno Rizzuti, who is a master, very experienced researcher in molecular simulation and computational tools. Thank you for your attention. Sorry, can I add? I forgot to add something. So this is a nice example that if you have a protein target that is interesting from the biomedical point of view, you can, I cannot promise you will find a drug, but I can promise you that you may find small compounds binding to that protein target. And through iterative process for optimization of the binding affinity, toxicity, and so on, you will be able to reach a promising compound that, with a bit of luck or a lot of luck, could reach the market eventually. Thank you very much. Sorry.